Somewhere right now, in a small clinic you have probably never heard of, a nurse is pressing a tiny device against a patient's arm, no bigger than a smartphone, and within minutes, before the patient has even finished their coffee, a readout appears showing whether that person's cells carry a mutation linked to cancer, whether a prescribed medication will heal them or harm them, and whether a disease their grandmother died from decades ago is quietly written into their own genetic code. Not weeks from now. Not after samples are shipped across the country to a specialized lab and processed by machines the size of refrigerators. Minutes. This is not a hypothetical scene from some distant future. It is happening today in hospitals, in disaster zones, in remote villages without electricity, in the palm of a researcher's hand on a mountainside thousands of miles from the nearest hospital. And it raises a question that should stop you in your tracks. If we can now read the deepest code of human life almost as easily as checking a text message, what happens next? What happens when the ability to decode a human being becomes as ordinary, as fast, and as cheap as taking a photograph? To understand how astonishing this moment truly is, you have to go back and remember what reading DNA used to require. Not centuries ago, decades. In the early 1990s, an international coalition of scientists launched one of the most audacious undertakings in the history of science, a project so ambitious that many researchers privately doubted it could ever be finished. The goal was simple to state and staggering to attempt. Read every single letter of the human genetic code, all 3 billion base pairs of it, and produce the first complete map of what makes a human being human. It was called the Human Genome Project, and it would take 13 years, cost roughly $3 billion, and require the coordinated effort of laboratories on multiple continents working around the clock. When it was finally completed in 2003, the achievement was celebrated as one of the greatest scientific triumphs in human history, on par with landing a person on the moon. And yet, within just 2 decades, something almost unbelievable happened. The same task that once took 13 years and billions of dollars can now, for certain applications, be completed in a single day for a few $100s. Not a typo, a single day. This is not a minor improvement. This is one of the fastest, most dramatic collapses in cost and time for any technology in recorded history, outpacing even the legendary exponential growth of computer processing power that gave us the smartphone revolution. The engine behind this transformation has a name, and it deserves to be far better known than it currently is. Next-generation sequencing. Unlike the older method developed by Frederick Sanger in the 1970s, which read DNA one fragment at a time in a slow, methodical, single-file process, next-generation sequencing machines shatter DNA into millions of tiny pieces and read all of them simultaneously, in parallel, like an enormous library where thousands of readers scan different pages of the same book at the exact same moment, and then a computer reassembles all those fragments back into the correct order afterward. The complexity of that reassembly process alone is staggering. Imagine shredding an encyclopedia into millions of tiny scraps of paper and then asking a computer to figure out, purely from overlapping fragments of text, exactly how the entire encyclopedia originally read without ever seeing the finished book. That is roughly what modern sequencing software accomplishes every single time a genome is decoded, and it does it with breathtaking speed and accuracy, processing what used to take a small army of scientists years to piece together manually. This technological leap did not happen by accident, and it did not happen without extraordinary failure along the way. Early attempts to miniaturize and accelerate sequencing technology in the 2000s were met with skepticism from established scientists who believed the existing methods, however slow, were simply the price of accuracy. Startups burned through funding, trying to build machines that could sequence DNA faster, only to produce results riddled with errors that made the data useless for medical diagnosis. One particularly ambitious approach involved threading a single strand of DNA through a microscopic pore and measuring subtle changes in electrical current as each genetic letter passed through, a concept called nanopore sequencing that sounded almost like something from a physics fantasy rather than achievable engineering. For years, critics dismissed it as impractical, plagued by noisy, unreliable signals. But the scientists and engineers refining the technology kept pushing, kept failing, kept adjusting, and eventually produced sequencing devices so small and portable that researchers have now used them to sequence DNA aboard the International Space Station in the middle of the Amazon rainforest, and in real time, during actual disease outbreaks, plugging a device the size of a candy bar directly into a laptop to identify a pathogen's exact genetic signature within hours of collecting a sample, something that would have been unthinkable even 15 years earlier. That capability was not just an academic curiosity. It became a matter of life and death. During major infectious disease outbreaks in recent years, portable sequencing technology allowed researchers on the ground to identify the exact genetic strain of a virus spreading through a population in near real time, tracking how the pathogen was mutating and moving from person to person, information that public health officials used to make urgent decisions about containment and treatment while the outbreak was still unfolding, not months afterward, when the data would arrive too late to matter. This is one of the quiet, underappreciated truths about the future of DNA testing. It is not simply about individuals learning more about their own bodies. It is about entire societies gaining the ability to see biological threats as they emerge, rather than after the damage is already done. But perhaps nowhere is the transformation more personal, more emotionally charged than in the realm of medicine, where rapid genetic testing has begun rewriting the rules of what doctors can diagnose and how quickly they can act. Consider the agonizing reality that used to define what physicians call diagnostic odysseys, cases involving newborn infants with mysterious, severe symptoms that didn't match any known condition, where parents and doctors alike would spend months, sometimes years, running test after test, watching a fragile infant suffer while searching desperately for an answer that might never come. Traditional genetic testing in these cases could take weeks to return results, an agonizing wait when a critically ill newborn's condition might be deteriorating by the hour. In recent years, hospitals have begun deploying ultra-rapid whole genome sequencing specifically for critically ill infants, technology capable of sequencing an entire newborn's genome and identifying a potential genetic diagnosis in under 24 hours, sometimes in as little as 13 hours from blood draw to diagnosis. For parents living through the most terrifying moment of their lives, that speed is not a technical achievement. It is the difference between a treatable condition caught in time and a tragedy that might have been prevented. Physicians using this technology have reported cases where an infant's genetic diagnosis, delivered within a single day, allowed doctors to immediately adjust treatment, avoid harmful interventions. The child's specific condition made dangerous, or in some heartbreaking cases, allowed a family to understand what was happening to their child and make informed decisions about care during their final hours together, a kind of clarity that simply did not exist a generation ago. This same acceleration is reshaping how we think about cancer, a disease that genetics has fundamentally transformed our understanding of over the past 2 decades. Cancer, at its core, is a disease of the genome, arising when mutations accumulate in a cell's DNA, disrupting the normal genetic instructions that control how and when a cell divides. For most of medical history, cancer was treated primarily based on where in the body it appeared and how it looked under a microscope. Today, oncologists increasingly treat cancer based on its precise genetics. Fingerprint, using rapid sequencing technology to identify the exact mutations driving a particular patient's tumor and matching that genetic profile to targeted therapies designed to attack those specific mutations, sparing healthy cells the devastating collateral damage of traditional chemotherapy. 2 patients with what looks under a microscope like the identical type of cancer might actually be fighting genetically distinct diseases requiring completely different treatments, and only genetic testing can reveal that crucial difference. Liquid biopsies might represent another leap forward in this space, a technique that allows doctors to detect fragments of tumor DNA circulating in a patient's bloodstream through nothing more than a simple blood draw, potentially catching a cancer's return months before it would become visible on a traditional scan, or monitoring in real time whether a treatment is actually working at the molecular level, long before physical symptoms would reveal success or failure. And this is where the story of DNA testing begins to intersect with an entirely different and rapidly advancing frontier: the ability not just to read genetic information, but to predict it before a person is ever born, or even before they are conceived. Prenatal genetic testing has advanced to a point where expectant parents can now screen for certain chromosomal conditions using nothing more than a simple maternal blood sample, detecting fragments of fetal DNA that naturally circulate in the mother's bloodstream during pregnancy, eliminating the need for far riskier, more invasive procedures that were standard practice for generations. Meanwhile, carrier screening allows prospective parents to learn, before ever starting a family, whether they each carry recessive genetic mutations that, if both partners carry the same one, could result in a serious inherited condition in their children, information that in previous generations families would only discover after tragedy had already struck. These advances have brought immense relief and empowerment to countless families, but they have also opened profound ethical questions that societies are still grappling with, questions about what kind of genetic information parents should have access to, how that information might influence deeply personal reproductive decisions, and where the line sits between using genetic knowledge to prevent suffering and drifting toward a society that quietly begins to value certain genetic traits over others, a concern rooted in a dark history of eugenics movements from the 20th century that were themselves justified, disturbingly, by the primitive genetic science of their era. This tension between extraordinary capability and genuine ethical peril runs through nearly every corner of the genetic testing revolution, and nowhere is it more vivid than in the emergence of gene editing technology, capable of not just reading DNA, but rewriting it. CRISPR, the revolutionary gene editing tool adapted from a bacterial immune defense system by researchers Jennifer Doudna and Emmanuel Charpentier, work that earned them the Nobel Prize in chemistry, has moved with astonishing speed from laboratory curiosity to clinical reality. In recent years, CRISPR-based therapies have received regulatory approval to treat certain severe genetic blood disorders, offering patients who endured a lifetime of pain, hospitalizations, and limited treatment options. Something that sounds almost too good to be true. A one-time genetic edit capable of functionally curing a condition that was, until that point, considered permanent and unmanageable, except through symptom control. Clinical trial participants who received these treatments have described a transformation so profound it is difficult to fully convey. Patients who had suffered excruciating pain crises since early childhood suddenly living free of them, sometimes for the first time in their entire lives. It represents one of the clearest, most emotionally powerful proofs that genetic medicine has crossed from theoretical promise into tangible, life-altering reality. Yet the same tool capable of curing devastating disease carries the theoretical capacity for something far more unsettling, and scientists across the world know it. If you can edit a gene to eliminate a disease, the same fundamental technique could, in principle, be used to alter genes associated with traits that have nothing to do with illness, est al., opening the door to what has become known, sometimes controversially, as the specter of so-called designer babies, genetically modified before birth not to prevent suffering, but to select for traits parents might simply prefer. This is not idle speculation. In 2018, the announcement that a scientist in China had used CRISPR to edit the genomes of human embryos, resulting in the birth of twin girls with permanently altered DNA passed down through the germline to any children they might someday have, sent shockwaves through the global scientific community, triggering swift, near-universal condemnation from researchers who viewed the experiment as reckless, insufficiently tested and conducted without appropriate ethical oversight or scientific consensus. The scientist involved was ultimately sentenced to prison in China, but the episode permanently changed the conversation around gene editing, forcing an international reckoning about where, exactly, humanity should draw the line between healing and enhancing, between curing disease and engineering. Preference, a line that the scientific community continues to actively debate today, with most major scientific and medical organizations maintaining a firm, cautious stance against heritable human genome editing until far more is understood about its long-term safety and its profound societal implications. Saliva sample to accompany in exchange for insights into ancestry, distant relatives, and have been used in ways their original users never anticipated. Law enforcement agencies have used public genetic genealogy databases to solve decades-old cold cases, identifying suspects not because the suspect's own DNA was ever in a criminal database, but because a distant relative, sometimes several branches removed on the family tree, had voluntarily submitted their own DNA for an ancestry test years earlier, creating a genetic breadcrumb trail investigators could follow. It is an extraordinary use of technology that has brought closure to grieving families after decades of uncertainty, identifying perpetrators of horrific crimes that had gone unsolved for a generation. But it has also sparked intense debate about genetic privacy, informed consent, and whether an individual's decision to learn about their own ancestry should carry the unintended consequence of exposing relatives who never agreed to have their genetic information used in a criminal investigation at all. And yet, for all the ethical complexity swirling around genetic testing, the trajectory of the science itself continues to push toward a future that sounds, on its surface, almost utopian. Researchers are actively working toward a world where every newborn might have their genome sequenced at birth as a routine part of standard medical care, creating a lifelong genetic health record that could flag disease risks decades before symptoms ever appear, allowing preventive interventions that could dramatically extend healthy lifespan. Pharmacogenomic testing, which analyzes how an individual's specific genetic makeup affects their metabolism of medications, is increasingly being integrated into routine prescribing decisions, allowing doctors to select not just the right medication for a diagnosis, but the right medication and dosage for that particular patient's unique biology, reducing the staggering number of adverse drug reactions that occur every year when patients are prescribed medications their bodies simply cannot process safely. Some researchers envision a future where genetic and epigenetic monitoring becomes a continuous, ongoing part of healthcare rather than a single test performed once, tracking subtle molecular changes in real time that might reveal the earliest, most treatable stages of disease long before conventional symptoms would ever prompt a doctor's visit. fundamentally shifting medicine from a reactive system that treats illness after it appears to a predictive, preventive system that intervenes before disease ever takes hold. There is also a quieter, more understated revolution unfolding in agriculture and environmental science, where the same rapid sequencing technology reshaping human medicine is being used to identify crop diseases in the field within hours rather than weeks, to monitor endangered species through environmental DNA collected from soil and water without ever needing to observe the animals directly, and to track the spread of antibiotic-resistant bacteria through hospital systems in near real time, information critical to containing outbreaks that could otherwise silently spread among the most vulnerable patients. The same portable sequencing devices used to diagnose critically ill infants have been deployed to remote conservation sites to combat illegal wildlife trafficking, allowing rangers to instantly identify the species origin of confiscated animal parts, providing crucial evidence for prosecution in a fraction of the time such analysis once required. What becomes clear when you step back and survey this entire landscape is that the future of DNA testing is not a single, isolated advancement. It is the convergence of decades of painstaking, often unglamorous scientific labor, stretching from Sanger's meticulous manual sequencing methods in the 1970s, through Kary Mullis conceiving of PCR amplification during a late-night drive, through the international collaboration that decoded the entire human genome letter by letter over 13 grueling years, through the quiet, persistent engineers who refused to abandon nanopore sequencing even when critics called it impractical, through Doudna and Charpentier unlocking the gene editing potential hidden inside a bacterial immune system, all converging into this singular moment where a device small enough to hold in one hand can now read the deepest code of human biology in minutes rather than years. It is, in the truest sense, one of the most significant technological transformations in human history, arguably as consequential as the invention of the microscope, or the discovery of antibiotics, or the mapping of the periodic table, each of which fundamentally altered what human beings were capable of understanding and controlling about the natural world. But unlike a telescope pointed outward at distant stars, this technology points inward toward the deepest, most intimate code that defines who we are, who our children might become, and what diseases we may or may not be destined to face. And that is precisely what makes this moment simultaneously so thrilling and so profoundly weighty. We are living through the exact period in history when reading and editing the human genome transitioned from an impossible dream into an accessible, everyday reality, and the decisions being made right now in laboratories, legislatures, and living rooms around the world about how this power should be used. Protected and limited will shape not just the next decade of medicine, but potentially the genetic trajectory of generations not yet born. Every rapid diagnosis that saves a critically ill infant, every targeted cancer therapy that spares a patient the brutal toll of traditional chemotherapy, every cold case finally solved after decades of anguish, and every unresolved ethical question still being fiercely debated in scientific journals and courtrooms alike, are all threads of the same extraordinary story. Humanity learning, for the first time in its existence, to read the ancient code written into every cell of every living thing, and daring, cautiously, imperfectly, to decide what we should do with that knowledge now that we finally have it.